Hi, my name is Samantha, and today we will be going over the cell communication signaling in pancreatic cancer. The pancreas sits behind the stomach and makes digestive enzymes and hormones like insulin to regulate blood sugar. Pancreatic cancer develops when these cells mutate and grow uncontrollably, usually starting in the ducts. It's hard to catch early because tumors don't show well on scans, and it often spreads before diagnosis. It's also resistant to many treatments. A major pathway involved is the growth factor signaling route, where an outside signal activates a receptor, which triggers Cras and then the downstream proteins that drive cell growth. These two images show where the pancreas sits in the body, right behind the stomach and in front of the spine. The diagrams highlight how a pancreatic tumor forms inside the pancreas and how it can grow into the surrounding tissue. The key signaling components that activate Cras in pancreatic cells include the EGF and EGFR. The main pathway starts with growth factors like EGF, which act as ligands. Ligands are signaling molecules released by nearby cells, and they bind to specific receptors on the target cell surface area. In this case, the receptor is EGFR, a receptor tyrosine kinase. When EGF binds to EGFR, two EGFR molecules come together and form a dimer, and this dimerization turns on the receptor's kinase activity. Each EGFR then places phosphate groups onto specific tyrosines on the other receptor's cytoplasmic tail. These phosphorylated tyrosines serve as docking sites for the signaling proteins inside the cell. Once those proteins bind, the signal can move inward to activate Cras and branch into major downstream pathways. The EGFR Cras pathway shows how signals from the growth factor receptor EGFR activates Cras and drives cell growth. In a resting pancreatic duct cell, Cras sits at the inner membrane bound to GDP, which keeps it turned off. When a growth factor like EGF binds to the EGFR receptor on the cell surface, EGFR becomes activated through phosphorylation on specific tyrosines inside that cell. That phosphorylation doesn't stay at the membrane. Helper proteins such as SOS and GRB2 transmit the signal inward to Cras. Once the signal reaches Cras, it triggers a key switch. Cras releases GDP and binds GTP instead. This GTP-bound form changes Cras into its active state. Now active, Cras can engage downstream effectors and turn on major pathways inside the cell. Two of the most important ones are the NAPK pathway, which moves into the nucleus to activate genes that drive cell cycle entry, and the mTOR pathway, which boosts survival, metabolism, and protein synthesis. These pathways together create the coordinated response the cell needs when receiving a growth signal. The final step is shutting Cras back off. Cras has the built-in ability to hydrolyze GTP back into GDP, which returns it to its inactive form. The left diagram here shows the full range of pathways Cras can activate after it switches to its GTP-bound state. Besides the two major branches that are usually focused on, the MAPK and the mTOR, Cras also connects to several other signaling routes to carry out functions like endocytosis, inflammation, transformation, and invasion. The second diagram is a simplified version that highlights only the two dominant pathways, showing how they feed into the nucleus to drive transcription that supports cell growth and survival. In the correct mechanism, Cras stays inactive until a growth factor activates EGFR at the cell surface. Once that signal comes in, Cras briefly switches to its GTP-bound active form, turns on the RAF-MEK-ERK and P13-KAKT-mTOR pathways to support normal growth or repair, and then quickly shuts itself off by hydrolyzing GTP back to GDP. Its activity is short, matched to the signal, and fully dependent on EGFR. In the faulty mechanism, however, Cras can't efficiently turn GTP back to GDP, so it remains active even when no growth factor is present. This makes the pathway run independently of EGFR and sends constant growth and survival signals through the same downstream branches. The continuous activation pushes cells to divide uncontrollably, resist cell death, and develop traits that support invasion, spread, and metastasis. This is my bibliography. Thank you for watching.